has a faster heart rate, then that means the entire wave is shorter. And some people would have a slow heart rate, so that means this whole wave is very long. I mean, these are measured in terms of milliseconds, all right? So you need to make some adjustment. So they do a QT divided by square root of R. From R to another R is one heartbeat. So that's what they call QTC. QT being corrected by heart rate, all right? Now, if you look at the total QT study, they call TQT. Somebody called a thorough QT study, same TQT. Right? ECG is considered as a safety variable, and the main concern is QT prolongation. And QTC is a corrected QT. And TQT is designed with matching time points. Look at ICH E14. So let me describe, so that's your ECG curve, right? Let me describe one kind of TQT study design. This will be a 48-hour study. So the subject, so the participant will carry a Holter monitor. So you carry this monitor at specific, at pre-specified time. This monitor will do the QT for you, will calculate the QT. Or look at your heart rate, look at the ECG curve. So the 48-hour study start like that. If we say 8 o'clock in the morning of day 0, for example, the subject will carry this Holter monitor. So at 8 o'clock, it will measure your ECG and calculate the QT or QTC. All right. And at 8:20, it'll calculate another QT. 8:40, 9 o'clock, 9:20, 9:40, until 7:40 the next day, day 1. So this day 0, the subject is not on any drug. All right. And you got a 24-hour QT profile. And between 7:40 and 8 o'clock the next day, in day 1, the subject's dosed with a study drug. And then 8 o'clock, there's a ECG, you do the QT, 8:20, 8:40, 9 o'clock, until 7:50 of day 2. So you got a 48-hour kind of ECG. And you do a time matched. You look at the day 1, 8 a.m. QTC minus day 0, 8 a.m. QTC. And you do the 8:20, 8:40, you do this 24-hour sort of QTC difference. You can find the maximum difference for that subject. So if you have 20 subjects going through this study, you look at the maximum of these difference. Every subject gives you a maximum. The mean cannot exceed 5 milliseconds. And the confidence interval, the upper limit of confidence interval cannot exceed 10 milliseconds. If your drug were able to demonstrate this successfully, you say, all right, we don't have a QT problem. So this is pretty much I want to cover about phase 1. Thank you. Today we are going to get into let's talk about phase 1 or 2, but I think the most part of these of this set of slides is going to be about phase 2. My sort of my career research has been on phase 2 designs and those findings, and so I will spend more time on this topic. I always start talking about this drug development process because from my perspective, the entire new drug development is about finding the right dose. The process starting from drug discovery, and we'll go through non-clinical development, and then we'll go through clinical development, phase 1, phase 2, phase 3, and phase 4 post-market. The entire process is about finding the right dose. Let's start with drug discovery. From the discovery lab, I think the scientist, if they want to look at some compound that could potentially be a drug, they'll have to look at the what they call the concentration response relationship. So they start out probably like molecular level or tissue level or molecular level, cell level, tissue level, organ level, or even the whole animal. What they do is that from the pharmacology point of view, if you increase the concentration, if the response does not increase, then you don't have a drug. In other words, if you're experimenting a new compound, if this new compound has the activity that you like to see, then by changing the concentration of this new compound, you should see the change in responses. Let's start out with this example about beta blocker. As I mentioned before, if you want to lower systolic blood pressure, you may want to inhibit the activity of beta agonist. So in the lab, what they did is that they have a very high concentration of the test drug or the test compound, and they sort of diluted it into a lower concentration. In a way, you probably have the 100% concentration, you dilute to half, you got 50%, you dilute another half, you got 25%, so on and so forth. So you got all the different concentration, and then you put the tissue into the different concentration and see if the tissue would respond the way you hope it does. So at certain concentrations, if the concentration is very low, you probably don't see any activity. And as a concentration higher than the very low concentration, if it starts having activity, and then you keep increasing the concentration, you're probably having higher and higher activities. Sometimes in the discovery lab, the increase of activity as you increase the concentration could be very profound, very significant. And in the lab, if you see that activity, you say, "Wow, this is a good compound. We want to promote into further development." So that's from discovery as an example, looking at the concentration response relationship. And then you get into non-clinical development, and there are many activities in non-clinical development. I mean, after discovery, you go through toxicology and more PK formulation and all these kind of activities. One example would be the animal toxicity. So the toxicology department, they want to look at the potential toxicity of this compound from 3 dimensions, one dimension being duration. So they will have acute experiments, subacute, subchronic, and chronic. You start with 1 or few days, up to a week or 2 weeks. And once you get into a longer term, you got 13 weeks, 26 weeks, or 52 weeks kind of toxicology studies. So that's one dimension. The next dimension is species. You may have mice, rats, rabbit, guinea pig, dogs, and different animals. And the third dimension will be concentration. So at this stage, they're looking at the concentration in terms of milligram per kilogram. In other words, mice have smaller body size, so their body weight is lower. And then you dose each mouse with different concentration, you say milligram per kilogram of body weight. And then you increase the doses, and then you will eventually see they're going to have adverse events that may affect certain kind of organs like liver, kidney, or lung, or their eye, or different organs. You may start seeing problems with these animals. And of course, you look at different species, you look at different duration, but the key is looking at different concentration. And one of the important deliverables from the toxicology department is that they have to deliver a very low dose, so low that you don't observe any problem in any species of the animals you experimented. And such a dose, they will call it a NOAEL, NOAEL, which is non-observed adverse event level. So you're going to have a very low concentration level such that at this level, at different species you study, there's no toxicology, there's no problem, no events to any of the animals. Now this NOAEL is absolutely important from toxicology, because once you get into clinical trials, you're looking at the first dose in human, you want to start with a very low level, and the first in human dose, the guiding principle is to look at that NOAEL, and then you look at that level, you translate into human body weight. Remember, it's milligram per kilogram, and you sort of get into a human body weight, milligram per kilogram. And from that NOAEL, after translation, you're going to multiply it by a safety factor like 1/10 or 1/20, and that'll be the starting dose for human. All right. So now we got into clinical development. We start with phase 1. Okay. So in a very high level, looking at the phase 1, 2, 3 drug development, well, phase 4 will be post-marketing. That's after approval. But phase 1, 2, 3 is pre-marketing, before approval. We develop this new drug. The focus is really about getting the right dose, or getting the dose right for this new drug. For example, in phase 1, the objective, like I said, is about finding about PK, PD, and MTD. Now, MTD, maximally tolerable dose, is what? Is the upper limit of your dose range. All right. So you go through phase 1, you get MTD. You escalate the dose. You start from very low dose escalation, right? SRD, MRD, you move up to MTD. And then we get into phase 2. You want to bring the dose down from MTD down to minimum effective dose, and mean ED. All right. Now, phase 1, you can establish the upper limit of the dose range. And phase 2, you can establish the lower limit of the dose range, which is minimum effective dose. So you're going to have a range of doses. And based on your phase 1 and phase 2 findings, you get the hopefully about right range of doses. And then you select one or a few doses, you move into phase 3. And of course, phase 3 is long-term, large-scale confirmatory trials. So what you do in phase 3 is, for the dose or doses you selected based on phase 1, phase 2 information, you want to confirm it in long-term, large-scale trials so that you provide sufficient evidence to regulatory agencies so that they can approve this drug. All right. So that's your phase 1, 2, 3, the pre-marketing development. Now, if the drug is good, that means it's efficacious and it's safe, it will receive approval, and then you go through phase 4, post-marketing. There are many drugs, the doses need to be adjusted after approval. So after the drug is already on the market, you go through a phase 4 studies, you realize, well, either the dose is too high or the dose is too low, you have to make adjustment. So what I'm trying to say is that finding the right dose is one of the key elements in any new drug development, and that covers every stage of drug development. So let's start with phase 1. Of course, we touched on phase 1 in the previous lecture. All right, it goes through healthy normal volunteers, primarily looking for PK properties, and help recommend dosing frequency. Based on PK, you can sort of try to propose like once a day, twice a day, or something like that, dosing frequency. And you want to estimate maximally tolerable dose, MTD. And of course, sometimes you cannot establish MTD, you have to establish MFD, maximally feasible dose. So phase 1 by nature is dose escalation design or crossover design to sort of study the PK properties and to increase or to escalate the doses. Now, as we mentioned before, the drug development programs for oncology or non-oncology are basically different. The strategies, the approach are different, all right? So if we look at the life-threatening diseases, all right, if you're developing life-threatening diseases, the drugs usually are more toxic, and so may not be ethical to use placebo control, may not be ethical to recruit healthy normal volunteers. And so for oncology, phase 1 trials tend to be open-label, single-arm dose escalation study designs. So in oncology, I think we are focusing more on sort of escalating the dose up to some toxic dose, because in oncology, what they believe is that, especially in the chemotherapy, is that you want to have the dose high enough so that you can kill cancer cells. Now, if you can kill cancer cells, it can be toxic to your ordinary cells also. So you're trying to get to a dose that's high enough, kills cancer cell, but it's not going to have negative or large negative effect to the human for the rest of the tissues. And like I said earlier, in oncology studies, we use cancer patients in phase 1, and it's not ethical for placebo control because they're already having cancer, so you don't use placebo control. And in a phase 1 cancer trial, you want to define this DLT, dose limiting toxicity, to see if this event occurs, you would call it a DLT. And the objective is to get to a point where the probability of toxicity at maximum tolerable dose is not going to exceed gamma, which is a preset probability, where gamma is a target probability of toxicity. The most popular phase 1 oncology design will be a 3+3 design, right? The most widely used design in oncology. Subjects are assigned to in group of 3 subjects, 3 per cohort kind of things. If only 3 subjects on this current dose, then if you did not observe any DLT, then you escalate with another cohort of 3 subjects on the higher dose. If there's 1 toxicity, you add 3 on the same dose. And if you found 2 or more toxicity, you would say MTD is exceeded. If 6 patients on the same dose, then if at most 1 toxicity, then you add 3 to the next higher dose. If 2 or more toxicities, then you claim that MTD has exceeded. The estimated MTD is the highest dose level with observed toxicity rate less than 0.33. So this is a schematic chart. You start with 3, and there's no DLT, you escalate. No DLT, you escalate. And you saw 1 DLT, you add 3 more. If out of the 6, you have only 1, then you keep escalating. Whenever you got 2 or more toxicity, you go back down to say, well, that level is, where your MTD has been exceeded. So this is a very typical 3+3 design. All right. So in the previous lecture, we talked about phase 1 studies, mostly on the non-oncology studies. And in the previous few slides, we'll look at the oncology studies. And remember, we need to find the phase 1. The most important objective is to understand the PK profile and to find MTD. Now, PK profile, we know it's important because if you look at the half-life, you use a half-life to propose as to a once a day, twice a day, or 3 times a day drug, or the dosing frequency. So PK profile is important, because without the PK profile, you're not sure how you're going to dose patients once you get into phase 2. All right. Now, phase 1 is healthy volunteers, or for oncology, in many cases, the patients in phase 1 trials, they do not necessarily have the target cancer you want the drug to treat. So you may not be able to see a lot of the efficacy data in phase 1. Phase 2, you use patients with a target disease. All right. In other words, if you want lower blood pressure, you want to recruit phase 2 patients with high blood pressure. All right. If you want to treat like arthritis, then in phase 2 you're going to recruit arthritis patients. Now, the phase 1 and phase 2, the major difference, of course, being the experiment unit. I mean, healthy volunteer versus patients with the target disease. The way you look at phase 2 is that this is the first opportunity we get to study the drug efficacy. All right. The drug started from the discovery lab, where they look at pharmacology, they look at toxicology. They started with the discovery lab. They move into preclinical development, and then they move into phase 1. So after all of this effort, this may take years of effort, you have not seen efficacy yet. So what you do in the first phase 2 trial is you really want to come up with a proof of concept study. It's making a go, no-go decision, all right? Because that's the first time you get to see the drug efficacy. And if the drug does not work, you stop developing it. So how to design the first phase 2, the first POC study? The traditional way of it is to do a 2-group design. Right? You have a test drug versus a placebo. So now here's a question: what dose of test drug you should use? Should we consider a low dose, or should we consider a high dose to compare with placebo? So let's think about it. So the objective is to make a go, no-go decision. If a low dose compared to placebo, the test drug doesn't work, are you comfortable to make a no-go decision? No, you're not. So what you want to do is, in the first POC, first phase 2, you want to use the highest possible dose. If the highest possible dose against placebo, you don't see any efficacy, then it's a comfort no-go decision, right? You say, well, given the best opportunity for this test drug, it didn't show any drug efficacy, so we stop developing this candidate. So it is critical at this stage to know that, I mean, the design thinking is that how to make a go, no-go decision. All right, only when the high dose is efficacious, then it makes sense to continue to develop the drug. And then you start looking at dose range, you try to see lower doses, responses and stuff like that. So that's why it is absolutely critical for phase 1 to deliver a good estimate of MTD. Now, in my experiences, there are many times MTD is either overestimated or underestimated. Now, if phase 1 MTD is overestimated, what would be the problem? Right? So suppose your true MTD is 100 milligram, but the phase 1 says it's 200 milligram. So you get into phase 2, you want to design a POC, use 200 milligram against placebo. what will happen? You're going to see a lot of adverse events, right? So that's not what you really want to see, because from phase 1, you're supposed to deliver an appropriate MTD. But overestimation of MTD is a less critical problem. The real problem is an underestimate MTD. All right. Let me give you an example. If the true MTD is 200 milligram, but the phase 1 told you is 100 milligram. Now you design a POC. You do 100 milligram against placebo. Now the results came out. It looks good, right? 100 milligram works. It's very safe. When you look at the efficacy results, you would think we can probably be able to get a better efficacy. All right, and 100 milligram is still very safe. If 150 milligram at a higher dose, we could be able to get a better results than what the 100 milligram delivers. So after discussion, you design another POC with 150 against placebo. So after the POC results came out, 150 works better than 100. And it's still very safe. And the team looked at these data and said, you know, if we increase the dose a little bit more, we may be able to get to see a better result than 150. And you may consider design the 200 against placebo. So what's wrong with this picture? All right. Now, in commercial drug development, the main reason the company spent billions of dollars to develop this drug is to hope that if the drug is efficacious and is safe, you can market this drug and generate a lot of revenue. But the amount of time you can generate revenue is limited by the patent life. Now, the patent life is 20 years from the day of application. So from the discovery stage or early development or non-clinical development stage, you have to file a patent because you don't want the other companies to follow your drug, right? So you want to have the patent. Now, once you apply the patent, then the clock is ticking. You have only 20 years. Now, a typical clinical development program takes, on the average, 8 to 12 years. All right. And so, in a way, you only have about maybe 10 years or fewer than 10 years to recuperate your clinical development cost, or drug development costs, including both preclinical and clinical. For a POC study, the way I described, it typically take about half a year from design until you see the results. Now, if you have to repeat the POC 2 or 3 times, then the amount of time you wasted is going to erode your patent life. So MTD, a good estimate of MTD or MFD from phase 1 is very, very critical. And the other important point I want to make is that how do we make a drug development process being most effective? All right. So the understanding of effectiveness in drug development is that if the drug is a good drug, we want the drug to reach the patient as early as possible. So that's what we want to achieve, no doubt. But as we know, the drug failure rate is very high. All right. It's probably, in general, I think from phase 1 onward is probably like 5 to 10% could be successful. Now, from the preclinical stage, from the discovery stage, it's probably only maybe 1 or 2% of the drug could be eventually successful. In other words, what I'm trying to say is that most of the drugs that we're developing will not be successful. So an effective drug development process would be, if the drug works, you want to reach patients as soon as possible. But if it doesn't work, you want to stop developing as early as possible. Right. So we can agree that this will be the most effective drug development process. But how can we achieve both objectives from our study design point of view? So the best way of drug development in terms of study design should be design to kill. Design to kill the drug, design to stop developing the drug. All right, so I design every study, want to make sure there's a clear no-go criteria. So if the drug cannot make it, let's kill it. Let's stop developing it. And this idea has been criticized to say in IT, this way you're going to kill every drug. And from my experience, there are many very good drugs. All right, if the drug is really good, there's no way you can kill it. I mean, you look at many of the marketed drugs, especially some of the market leaders, like Lipitor, Spreva, or many of these very good drugs are on the market. In the development process, it's very clear. I mean, the efficacy and the safety is very outstanding. There's no way you can kill it. So very, very few drugs are that good, but most drugs are not good. So if you can stop developing the drug as early as possible, then you make the development process more effective. All right, because if I design to kill, every study is a no-go study. All right, so this way, if the drug is not good, it's not good enough, it will not be moving forward. All right, so POC is a very good example of design to kill. Now, why would I say that? In this first phase II trial, instead of doing a placebo-controlled trial, you can consider an active control trial, for example. All right, you say test drug against the market leader. If the test drug is not inferior to the market leader, we can move on. All right, yes, but if the test drug is inferior to the market leader, it's not clear kill. Because you say it may not be good enough, but it may be good in other aspects. So it's not a clear go, no-go decision. But for placebo-controlled study, if it doesn't beat placebo, it's a clear kill. It's a clear no-go decision. All right. So if you think about drug development process, the most effective way of developing any new drug is to design to kill. All right. So this POC is absolutely critical. And I will spend more time in this lecture talking about POC. POC is very, very difficult. So the two objectives in phase II are POC and dose ranging. Remember, the dose ranging part is about moving the efficacious doses low in a hope we can find the minimum effective dose, which is the lower limit of the dose range. So the two steps are POC and dose ranging. All right. So phase II objectives is to propose dose or doses for phase III design, because if you can establish the lower limit of the dose range, then you can recommend one or two or few doses for phase III studies. The key is moving doses down to minimum effective dose. Now, if dose range is not found in phase II, it will be too expensive in later phases. Let me give you an example. Now, this company was able to successfully market a drug at 100 milligram in Europe and in many other countries. So 100 milligram was already approved in many countries, and they were marketing it at 100 milligrams. And this company wanted to bring this drug to the United States. Now, so FDA looked at their package and said, look, 100 milligram looks good. Efficacy is good. But we would like to see the efficacy and safety at lower doses. So the company went back and designed the dose ranging trial with doses below 100. So what they did was placebo, 25, 50, and 100. And the 25 milligram worked. So FDA says, okay, we'll approve 25 milligram. So what's wrong with this picture? So we need to have some understanding about the drug pricing and about the drug marketing, all right? Now, in most countries, the drug price is linear to dose. In other words, if 100 milligram is $1, 200 milligrams is $2, 300 milligrams is $3. All right. With the exception of United States. In United States, the drug price has nothing to do with the dosage. So it's a free market. So the company can price their drug whatever way they want. But in most other countries, the drug price is linear to dose. So in that sense, the marketing strategy is you'll always price the drug based on the lowest approved dose. So this company sort of marketed 100 milligrams in many countries with the 100 milligram price as a base price. Okay, now if FDA approved 25, then every country is going to lower it to 25. So this company's revenue on this drug will drop 75%. This is a huge mistake. All right, so when I mentioned this story and people says, well, Nike, this is a commercial issue. This is not a scientific issue. And I say this is an absolutely scientific issue because the only time, the only opportunity you can find the right dose is phase 2. If you missed it, if you didn't get the right range of doses at phase 2, you had to change your dose strategy at phase 3 and phase 4. It'll be very, very expensive. I may not be able to get to my examples in this lecture, but in future lectures, I will have many examples about finding the wrong dose and the consequences. POC, all right. Typically 2 treatment groups, parallel design, placebo control, use a dose at MTD or very close to MTD. So you have a high dose against placebo. Short-term clinical efficacy endpoint, sometimes use surrogate markers and moderate sample size. All right, so that's a typical POC. And I hope to tell you that POC looks simple, right? But it's very, very, very difficult. Okay. So the team come to me, the statistician, asking about sample size. So how do we calculate sample size for POC design? All right. Now this is the opportunity for a statistician to contribute to the study design. So this is my opportunity to work with the team to educate the team about the risks the team is willing to take. And we have to really drill down to the assumptions that the team is making. All right. Now, of course, we know sample size calculation is about alpha, beta, delta, sigma. Let me slow down a little bit. Alpha, beta, delta, sigma. Alpha is probably making a type 1 error. Beta is probably making a type 2 error. Delta is a treatment difference, the expected treatment difference. and sigma being standard deviation. All right, so we have 4 quantities to determine the sample size. Now, if I want to simplify it, I would divide the delta by sigma. So once I divide delta by sigma, I end up with variance equal to 1, sigma equal to 1, right? Delta divided by sigma. So I'm trying to calculate the sample size just based on delta over sigma. So if you know delta, and if you know sigma, then sample size becomes a function of alpha and beta, right? So in the following example, I'm going to assume delta being positive. Maybe this is for COPD, all right? Chronic obstructive pulmonary disease. In COPD, they're measuring the FEV1, which is a forced expiration volume. They have this device. You blow into the device, right? Once you blow into the device, the device will measure the volume out of your lung when you exhale, and so the volume increases, but at 1 second there's a particular volume, and this is FEV1, forced expiration volume at 1 second. All right. So when you compare test drug against placebo patients, they're COPD patients, their lung function is deteriorating, right? And placebo doesn't provide any help. So their FEV1 tend to be low, or changing FEV1 from baseline to endpoint. Now, the test drug should have a higher FEV1. So here, if the test drug provide more FEV1, now delta is positive. So delta is the difference between test drug and placebo. So your test drug mean subtract the placebo mean is a positive number. All right. So in the later illustration, I'm going to use these assumptions. Assuming delta is positive and assuming variance equal 1. POC fundamentally is a hypothesis testing problem. All right, it's about alpha control. Now, primary endpoint is clinical efficacy. And pre-specified two-sided alpha could be greater than or equal to 0.05. And power may be greater than 80%. And you want to take this opportunity to make a go, no-go decision. All right. So people said, how come you designed the study with alpha greater than or equal to 0.05? In my experience, I used to design studies, POC studies, with alpha being 0.2. And if you look at publications, there are studies designed with alpha being 0.4. And people say alpha 0.4 is almost flipping a coin. How can you design a study with such a high risk? All right. And when I make a presentation about my study with alpha being 0.2, I was challenged by the audience to say, does alpha really matter? I say alpha matters a lot, as we looked at in the first lecture. Alpha is absolutely important. And then they say, what do you mean by alpha being 0.2? I say, alpha being 2 is that if you got a p-value after the POC study, you got a p-value of 0.19, I would say statistically significant. If you got a p-value of 0.21, I say it's not significant. And the question is that why can you make alpha being 0.2? So here's why. Now, what is type 1 error? All right, type 1 error is the risk of developing a placebo. So what is a type 2 error? Now, type 2 error is a risk of giving up a good drug, right? So type 1 and type 2 are different risks we're going to deal with. Now, remember, a POC is about making a go, no-go decision. All right, so this is my first phase 2 trial. The first time this test drug is going to be tested in patients with a target disease. And I need to make a clear go, no-go decision. But either decision, I could be making a mistake. All right. I can make only one decision. I cannot make two decisions. There's nothing in between. I cannot say, well, it's go or it's no go. No. You have to make a clear decision. After POC, you have to go or you're going to stop development. You have only one decision. But either decision, you may make an error. All right. But for a given decision, you can only make one type of error. You will not be able to make another type of error. In other words, that decision can be either correct or can be wrong. All right. So if you make a go decision, if it is correct, you're fine. If the decision is wrong, you're making a type 1 error. It's an alpha risk. If you make a no-go decision, the decision could be right. The drug is not good. Or you could make only a type 2 error. Now, the example I want to give you is if the team came to me, says, "IT, we want to do a POC design, and we need your help to calculate sample size." So we started having a conversation. And then they said, by the way, because the budget is limited, we can only afford 100 patients. So I'm a statistician. I'm supposed to calculate sample size, but now you tell me my sample size cannot exceed 100. So I have to understand the alpha, beta, delta, sigma, right? So they gave me the alpha, beta, delta, sigma. I came back and did the calculation. I said, I need 800 patients. And they say, no way. I mean, the budget is limited. I mean, there are many reasons why a POC cannot afford too many patients. Budget is one of the reasons. The other reason is that from an ethical point of view, you don't know the drug works or not. It may not be ethical to expose this test drug with too many patients. So 100 patients is reasonable, okay? So now I said, but given alpha, beta, delta, sigma, I need 800 patients. And now we sit down and discuss, can I increase delta? All right. Now, remember, this is the first time we get to see the test drug in patients. So before POC, or at the time of designing POC, we don't know the drug efficacy. So you can only guess a delta. There's not such a delta associated with this drug. You don't know that. So how do you guess a delta? There's 3 ways of guessing delta. The first way is to guess the delta with other drugs. All right, other drugs of the same class. You look at the approved drugs and the market drugs to see how much the difference that those drugs delivered compared to placebo. So that's one we're looking at from other drugs. The second way is to look at from the drug itself by looking at the preclinical data, look at the animal models, and you try to extrapolate that delta from animal to human. So that's the second way. Now, the third way of guessing delta is a reverse engineer. In other words, by keeping your sample size as a limit, and you reverse calculate a larger delta. So I communicated with the team, and I said, if we have to do a 100 patient trial, then I need to increase the delta. So we started discussing delta. How much delta can I increase? So if I keep pushing Up to a point, they say in 90, there's no way any drug could deliver that kind of treatment difference. You know, this delta is the biggest possible delta we can ever afford, or we could ever see in practice, in reality. I say fine. So now we have a delta. We have a sort of agreed assumed delta, and then come back and calculate sample size. I still need 300 patients. No, but we can only afford 100 patients. So given the very large delta and given sigma is known, usually sigma is not a problem because you look at the publications, you look at, and most drugs that already, I mean, many diseases, we know the sigma pretty well. All right, so sigma is not an issue. Delta is a big issue. But you come up with a delta that's agreeable. And I want to reduce the sample size from 300 to 100, then I have to increase alpha, increase beta. So I increase alpha to be 0.2 to make the study feasible. All right. Now, all right, yeah, you're making the study feasible, but is alpha 0.2 a reasonable approach of designing a POC study? So let's come back and look at the picture of the entire drug development process. The entire drug development process, of course, from the basic science at the discovery lab, they have this understanding of the disease and understanding the target and all that kind of stuff. But once you get into human testing, we don't know the drug is good or not for human, all right? So you can think of it as a gambling process. So the company is investing their resources to gamble to see whether the drug works or not. So think of you as a company and sitting in front of the poker table, all right? So you sit in front of the poker table, you have to put down some chips, and the dealer will give you 1 or 2 cards or something like that to begin with. You get a few cards. If the cards looks good, you put down more chips. All right. This POC is when you first sit down and you only have limited chips and you want to buy the first few cards to look at. So with a large alpha, that means you have smaller sample size. That means you have limited chips. You want to buy the card. That does not mean you didn't get an ace, right? But you have to put on the chips before you get to see the first card. That's your POC design. You have to come up with a sample size and see if the drug works, if there's any hope to further develop this drug. So a few chips doesn't mean you don't have a good card. Now, remember I told you about this study design was alpha being 0.2? When the study results came out, the p-value is less than 0.0001. There's no argument. It's a clear go decision. But if I don't go with the first design, I mean, not even be able to see how well the drug works. So with 100 patients, I'm just buying my first card. All right. Now, of course, we know the danger of this being your alpha is too big, your beta is too big. It's very easy. You can make a wrong decision. Yes, because your precision is slow, right? With smaller sample size, you don't get a lot of good precision. And it's our job as a clinical statistician to explain to the team so that the team can understand the risk they're taking. And to me, sample size reflects the entire study design. Why? Because sample size is based on alpha, beta, delta, and sigma. All right, what's alpha and beta? Alpha and beta are the risks the team is willing to take. The delta and sigma are the assumptions the team is willing to make. So without the assumptions, without the risks, we cannot calculate sample size. Once I come up with a sample size, the sample size reflects the risks the team is willing to take and the assumptions the team is willing to make. And I see, based on my years of experience working on clinical trials, most of statisticians are struggling with communication. So I'm going to provide a communication tool to help clinical statisticians how to communicate with the rest of the team so that the non-statisticians understand what we're talking about. So what I'm saying here is that the POC design, even though it's only 2 treatment groups, but it has to help the team make a go or no-go decision. Okay. Now, let's look at, let's see. I'm not sure whether I have enough time to cover this communication tool today. Let me start out this communication tool. If I cannot finish in the next lecture, we'll continue. Okay. So this discussion should happen before breaking blind and after the design is finalized. And we have to set clear go, no-go criteria. All right. Now, why it has to be before breaking blind? So we're talking about probability. All right. Now, probability will only exist before you see the data. The easy example of being flipping a coin. All right. Before you flip the coin, you would say the probability of getting a head is 1/2. The probability of getting a tail is 1/2. So now flip the coin now landed. Once the coin is landed, you're going to either see a head or tail, right? There's no probability now. You see your observation, you see your data. All right, so probability can only happen before the coin is landed. Now in clinical trial, when did the coin land? When you unblind. Once you unblind the trial, the coin is landed. There's no probability. So if you're talking about alpha or beta, the probability of making a type 1 or type 2 error, you can only talk about it before you see the data. All right. Once the trial is unblinded, the data is out there, there's a p-value. The p-value is nothing more than a descriptive statistics. Because given the pre-specified analytical methods, given this data set, you're going to get a p-value. That p-value does not represent any risk. It's just a p-value. It's just an observation. But the risk being you're making a wrong decision. You look at the p-value, you're going to make a decision, right? You make a go decision, you make a no-go decision. But either decision, you may have a mistake. And the risk of making a mistake can only be communicated before the coin is landed, right? And before the blind is broken, right? And I say this communication tool is only applicable after the design is finalized, all right? Remember, I said this case was only 100 patients. So I have only 100 patients. I finalized this design, and now the study is ongoing. And then I come back and talk about this communication tool and talk about the go, no-go criteria. All right. If I didn't do it before the design is finalized, the team can always change their mind. All right. By looking at the communication tool, they may say, well, in that case, we may increase to 120 patients. And then you have to redo the whole discussion, the whole design kind of stuff. So I think I may not be able to start the communication tool today. Let's stop here at slide number 13. We'll start talking about the communication tool in the next lecture. Thank you.